குட் மார்னிங் ஸ்டூடெண்ட்ஸ். நம்ம ஆஃபல் ஏவியேஷனுக்கான சாப்டரில் 5th சாப்டர் பார்க்குறோம். அதில் ஜெனட்டிக் கோட் பார்க்கலாம். DNA is a genetic material that carries a genetic information in a cell and from generation to generation சொல்லலாம். At this stage, an attempt will be made to determine the what manner of the genetic information exists the DNA molecule. Are they written the coded language and the DNA molecule? If they occur in the language of code, what is the nature of the genetic code, and translation of the protein follow the triplet rule and sequence of the 3 mRNA bases codon designates the 20 different kinds of amino acids used in the protein synthesis, and genetic code is a sequential relationship between the nucleotide gene and amino acid in the protein they encode. There are 64 possible triplet and the 61 of them used to represent the amino acid. The remaining 3 triplet codon and derives the signals for polypeptide chains. Since there are only 20 amino acids involved in the protein synthesis, most of them are encode more than one triplet. Two things make this multiple coding possible. The first 3 is more than one tRNA for most amino acids. Each tRNA has different anticodon. Second is pairing is highly specific for the first 2 proton portions on the codon, permitting Watson-Crick base pair to be formed. But the third position, there is great deal of flexibility as to which base pair are acceptable. Most part of the genetic code is universal, become being the same prokaryote and eukaryote also. The order base pair long DNA molecule control the kind of order of amino acid found in the protein of an organism. The specific order of the base pair called genetic code, but the blueprint is establishing the kind of proteins to be synthesized which make in the organism is unique. Marshall Nirenberg and Severo Ochoa, the enzyme polynucleotide phosphorylase is also called Ochoa enzyme, and Hargobind Corona and Francis Crick and many others have contributed to the significance of the disappear of the genetic code. The order is basis of arranged in mRNA decide in the order in the amino acid are arranged in the protein. Finally, the checkerboard of the genetic code is prepared. அதை நம்ம டேபிளேஷனில் பார்த்துக்கலாம் இல்லையா. இந்த salient feature of genetic code very important 5 mark. The genetic code is triplet code அப்படின்னு பார்த்தா 16 codon for the amino acid and 3 codon not code any amino acid as a function பார்த்தோம்னா stop codon அதை terminate codon சொல்றோம். The genetic code is universal அப்படின்னு பார்த்தோம். பொதுவானது, உலகத்துக்கு பொதுவானது. Means that all known as living system use the nucleic acid and same 3 base codon, triplet codon, and direct in the synthesis of protein from amino acid. For example, the mRNA W codon codes for phenylalanine in all cell of the organism. Some exceptions also reported in the prokaryote, mitochondrial and chloroplast genomes. However, similarities are more than differences. Next, a non-overlapping codon means that the same letter is not used for 2 different codons. For instance, the nucleotide sequence guanine, uracil, guanine, uracil, cytosine represent only 2 codons. If a comma is less, it's also not necessary, which means that message would be read directly from one end to another. There is no punctuation needed for the between the 2 codes. Next, the degenerate codon means that more than one triplet codon could code for specific amino acid also. For example, the codon guanine, uracil, guanine, uracil, cytosine, guanine, uracil, adenine, guanine, uracil, and guanine for only represent the valine. Next, non-ambiguous code means that one codon will code for one amino acid only. No confusions. So the codon is always read for the fixed direction. That means for fifth and to third interaction, that's called polarity. So adenine uracil guanine has a 12 function. What is the 12 function? Act as the initiator codon and also code for the amino acid methionine. So 12 function is contained. Next, so adenine, uracil adenine guanine, and uracil guanine adenine codon designated the termination codon. This also nonsense codons without any meanings. I mean nonsense codon. Next, mutation and genetic code. We will discuss about the mutation and genetic code. And comparative studies of the mutations, the sudden change in a gene, particular gene, is corresponding the alteration of the amino acid sequence of the specific protein have conferred on the validity of the genetic code. The relationship between the gene and DNA are the best understood by the mutation studies also. The simplest type of the mutation at the molecular level is a change in nucleotide that substitute one base for another. So changes are known as base substitutions, which may occur spontaneously or due to the action of mutagens, as well as a well-studied example. Sickle cell anemia is a human is a result from the point mutation of allele beta hemoglobin. That means HB, beta HB. A hemoglobin molecule consists of four polypeptide chains of two types, two alpha and two beta chains. Each chain have a heme group and of its surface. The heme group are involved in binding of oxygen. The human blood disease, sickle cell anemia, is due to abnormal hemoglobin formation. The abnormality of hemoglobin is due to the single base substitution of the sixth codon of the beta globulin gene from GAG converted to GTG. So in the beta chain of the hemoglobin சொல்லலாம் is a result change of the amino acid glutamic acid to the valine sixth position of the beta chain. This is a classical example to point mutation that result in change of amino acid residue the glutamic acid to the valine. The mutant hemoglobin undergo the polymerization under the oxygen tension, causing change in the shape of the RBC from pi concave to sickle shape structure. The effect of the point mutation can be understood by the following example: A, B, C, D, E, F, G, H, I, J, K, L. For the interest insert in the letter O between the D, E, F, and H, the arrangement would be collapse. Okay, the sequence also changed. The insert of the O and Q, the same place also would be the reading position also completely changed. So the above information show the insertion of the or deletion of the one or two bases changes in the reading frame from point of the insertion of the deletion. These are deletions. Such mutations are referred to as the frame shift insertion or deletion mutations also. These forms of genetic basis have proof that codon is triplet and in continuous manner also is completely proved. Okay, students, next. The DNA is adapter molecule. The diagram also very very important diagram. Note the diagram and pause. Draw label the part of the tRNA molecule. Okay. Okay, thank you, students.